And now it's my pleasure to introduce Dr. Joanna Barbara. Joanna joined Xenotech in 2007. She served as Vice President of Scientific Operations from 2017 to 2022 when Xenotech was acquired by BioIVT, and now leads operations as the site head. Dr. Barbara obtained her PhD in analytical chemistry from the University of Florida and her MBA from the University of Kansas. And with that introduction, please allow me to pass the presentation over to Dr. Barbara.
Oh, thank you, Michael. Good morning, everyone. Okay, I'm going to start out by a little bit of background about me and how I fit into BioIVT, because as Michael said, I'm from Legacy Xenotech. So BioIVT is about 42 years old, and one of the things that we do at BioIVT is grow by acquisition. So here is a picture of all of the acquisitions over the lifetime of the company. At the time of making this, the most recent one was Precision Med in 2023. But in 2022, BioIVT significantly increased its footprint in ADME-Tox and purchased Xenotech and Cypex that year. I was part of Legacy Xenotech executive leadership team at the time, and so I stayed, and now I'm part of BioIVT. I fit into the ADME-Tox business unit. There are 4, but today we're going to talk a little bit about the in vitro ADME programs that we do. We do offer a huge variety of hepatocytes and subcell fractions and various products as well, but that's not where I'm focused this morning or this afternoon, as depending on if you're in Europe. So today we're going to talk about the in vitro ADME programs that we perform, specifically transporters, although we perform pretty much everything in vitro DDI related and metabolism related. So this summer we launched our research service tiers. I do want to share it because I'm excited about it and I'm pretty proud of it too. We did basically officially split our offerings into 3 different kinds of things to meet different customer needs, because we were finding it was taking a little time to initially understand what people needed. So we have a tier 1 basic program, which is useful for decision making. It's not really for submission. That's not its point. It's trying to determine if it's a liability situation. It's not really screening. It's more about when you've got 1 or 2 compounds and you're making decisions or plans. Screening is not our wheelhouse, so we typically don't go there very often, unless it's transporter screening performed at the Santa Clara lab, but that's not the focus of today. Tier 2 is our IND enabling program. And that is basically definitive data for IND submissions. It's very much designed for IND submission. And then tier 3 is the same sort of thing, except if you would like to have GLP compliance or want everything to be eCTG format ready straight off the bat, that's kind of where you would land. We also offer custom studies. And the reason I wanted to bring this up before we talk more about the actual scientific topic is because you will see in my case studies, we come in with a few different things. We actually come in with a tier 1 and a tier 2. We didn't call them that at the time, but where we landed was because the data was weird, we landed in custom studies. And that's something I really like doing. I really like the troubleshooting part of this. This is why I've stayed in the CRO space for 16 years. So we're going to talk about uncooperative drugs, which sometimes feels like all of them, but isn't. And we're going to focus a bit on what I like to think of as practical science. Usually the people we work with, the people we talk to, are all very thoroughly trained scientists. There's a lot of brilliant people I get to interact with in my job. But sometimes it's hard to translate that into, okay, what are you going to do to move this along and get to your business endpoint? Because at the end of the day, we're all trying to get these compounds to patients. And so we can explore a lot of things, but we need to make sure it's useful. So I'll start out by talking a little bit about our toolbox and some of the ways that we look at this kind of work, and then touch a bit on the ICH guidance, because that's a really big deal right now and pretty exciting. Then we're going to spend the bulk of the time on some case studies, various ones. Some of them are pretty fun. And I'm not going to talk about conclusions, because I always think it's funny to talk about your conclusions before you talk about the rest. So in vitro drug transporter assays are used for a lot of different things. In DDI work, which is what I'm focused on today, we're generally looking at cell permeability or inhibition and substrate potential. It ranges for us from more screening type approaches, pseudo screens, if you will, to really complex assays, depending on what the customers bring to us, because we're always working on other people's compounds and programs. That's one of the fun parts about what we do, actually. We see a lot of diversity. And when drugs have really good physicochemical properties, everything's fine and you can chug along. But that isn't most early drugs in development. The vast majority of them are pretty difficult and challenging to work with, which means that you have to really think about what you're doing to make sure you can draw conclusions and move forward. I don't know what this baby did, but I know that look on that lady's face. I feel that a lot. It usually begins with solubility. I'm going to take a little step back and talk about test systems, because that's the basis of our toolbox. And I do think of this as a toolbox. There are tons of transporter proteins. Something like 10% of the proteome, most proteomes, is actually transporter proteins. And we focus on a very tiny subset of those for DDI purposes because we know them well and we know where they're involved. But you're kind of bound when you're looking at these transporters by how you can look at them, what test systems are available to be able to actually look at each transporter. For us, we use a variety of them, and they usually fall into these categories. Actually, we have some really great opti-expression technology over in Santa Clara, where we can customize and build specific transporters. I'm not going to talk about that today, but it's really cool, and if you're interested in that, it's on the website. We typically in the KC lab focus more on the specific subset of DDI, because a lot of our work is looking at packages and trying to assess both enzyme and transporter liabilities for drug-drug interactions. So the issue is that the way that you do the assay is completely dependent upon what kind of test system you're using. And for us, predominantly in Kansas City, we're going to use KCO2, and we have to, usually for PGP or maybe some permeability studies. We will use MDCK2 in various ways, actually for a lot of different things across the board. But today I'm focusing on PGP and BCRP there. So we're talking about those efflux transporters. And then HEC 293 is the cell line we usually use at Kansas City for solute carriers. You don't have to, but in the case studies I'm going to share, that's what we were using. And then we also do some work in primary cells. All of the others are immortalized cell lines that somebody has worked on to make sure they overexpress the protein, the transporter protein of interest. We use them whenever we can because they're more specific. But we also use primary cells sometimes, which are hepatocytes for us, because we're usually looking at ATP 1B1 and 1B3. If we are looking specifically at that, and there's a reason, we typically don't start there because you don't have the benefit of the overexpression, so it can be hard to pull apart data from those. But if you want a bigger picture answer, then that's where we might go. And then the other thing that we use is bits of cells. That's how I think about it. And for this particular work, it's membrane vesicles. For enzyme work, it's more like subcellular fractions, microsomes, cytosol, that sort of thing. If we're using membrane vesicles, it means that we couldn't get a more complex test system to work on them. So maybe we need it for MRP2 or something, because it's hard to find a lot of options for test systems for those. And so we validated some vesicle assays so that we can always work on those. Usually for us, we're going to use those for other ABC proteins. The challenge is that all of these different test systems need completely different conditions. They like different formats. They're happier in a different buffer, especially the cells, very finicky. And it kind of dictates the controls and the way you look at the data to be useful, which is interesting, but it's also challenging to think about as a whole. So we wouldn't expect, if you put a drug in two different solvents, we know inherently that we wouldn't expect the solubility to be identical. If you put something in water versus DMSO versus ACN, you would never think, oh, well, it'll be exactly the same. We know that. So we don't expect it to be unaffected by pH or buffer ionic strength or any of those other things either. And this becomes relevant when we're looking across the different test systems. So here is an example of a few of the different assay conditions we use. There's flexibility in some of these, not in all, depending on how they're validated, because we are a GLP compliant facility. So if we're going to use the validated assays, then we already have to have rock solid data and the infrastructure to do them. So our hands are a bit tied. Also, sometimes the cells won't work under other conditions or won't perform the way we'd like them to. So for KCO2 and MDCK2 assays, we typically are going to be doing a transwell format with what I'm going to call buffer A, because there's no need to get into the details of that, at pH 7.4, and we'll do various time points, but they're usually up to about 120 minutes max. In contrast, if we're looking at HEC293 cells, we're going to do a basic monolayer. We're going to just plate those, use buffer B, pH 7.4 for shorter time points. In this case, though, there are some caveats. For OATPs particularly, we always do a 30-minute pre-incubation, so that's another variable to add into this mix, and we include butyric acid. So yet another variable. If we're looking at mates, whatever the test system, we analyze those at pH 8.5, so that's another difference. Then vesicle assays have another set of buffers. So buffer C, D, E, depending on the transporter. So we have to deal with all sorts of experimental conditions. Yet at the end of the day, what we're trying to do is compare the data for PGP in the same kind of way as we're going to look at the data from OCT1, for example. So that can be a difficult thing. I'm going to do a very brief and recent history of regulatory guidance, because this is one of the ingredients in the work that we do that really is very important. What we are all trying to do is get these drugs to market. So we have to make sure that we can say things like, yes, we meet the guidance for the FDA or the EMA or the PMDA or whoever else is the target for this particular drug and particular customer. The interesting part about it, though, is that they all say very different things. And so for us, we often don't have control over where this goes once we're done with it. We don't have the flexibility to say, well, you know, your drug doesn't whatever, so we don't need to focus on this area. We have to be able to feed everybody. We have to be able to make sure that you have options. And we take it pretty seriously. One of the things that we like to do is have a talk about the compound and the program earlier on in this experience, so that we can make sure we're not, you know, having you meet all FDA guidelines, and then in 2 years you plan to submit to the EMA, but we missed a little difference there and didn't have everything ready. So we are trying to cover everything. A lot of them have something to say about supporting information, and this is where it gets very interesting. Some of the assays we do, you know, an IC50 is pretty straightforward. You know what you're doing. The challenge is the pieces that aren't necessarily visible. And so a lot of these guidances specifically call out these days cytotox and non-specific binding and that sort of thing. And they do differ under the various conditions, and that's why it's important. But the real challenge is that all of these typically say something different. So what we had to do, and we started really doing this in 2017, but we were already doing it a bit before. What we had to do is figure out, okay, so what's the most conservative approach that will meet all of the different guidances, so that we can be sure our customers have what they need going forward and aren't going to have to come back and redo something or fill a gap. Because a lot of the studies we see, it's because somebody missed something a couple of years ago. And so we're trying to make sure that our customers don't have to deal with that unless it's unavoidable. So just like all other science, and one of the reasons I love being in science is we get to learn as we go and build and improve and kind of stand on the shoulders of people that worked before us. And so this is true in this regulatory guidance scenario environment too. So over the years, people learned more and more. We gained more data, we understood more, we were able to look at things in a different way. And that's a double-edged sword though, because on the one hand, now you can really understand better what's happening with your drug. So you have a better shot at getting it all the way through to the patient. But the other side of it is it becomes quite big and you have to consider a lot of other things. And it becomes quite difficult to make decisions quickly. In 2017, the FDA gave the first really explicit list about things to consider in transporter research. And here is a list of it. I'm not going to read it to you. The challenge is not all of the guidance documents agree on what to do with them. This isn't only apparent in drugs and drug transporters. We also see it a lot with enzymes. We need to look at some of the different things that change how you think about the assay. The big one is often non-specific binding, and the other big one is cytotox. Those are the big ones that really can make a giant difference, and you have to consider pretty early. So these are things that we all have to pay more attention to because we understand they are going to make a difference to interpretation, potentially. Sometimes in silico estimates are fine, but sometimes you really need some data. Now, the ICH draft guidance, M12, came out in 2022, which was wonderful. CROs across the globe were very happy because now we don't have to keep trying to balance all of the different regulatory guidances from everybody else, because all of the big players are actually part of the ICH anyway, and so harmonization is in sight, which is wonderful. Something I really like about this guidance, though, is also it's kind of open to thinking and a considered approach. And it's not as prescriptive as some of the previous guidances because you have to have some flexibility for the compound and the program in question. So the ICH says that several factors can cause actual drug concentrations in the assays to deviate from what you thought. And some of the bigger ones are solubility, particularly, non-specific binding and instability. So if this is a big problem, basically it says there should be corrections. Well, we've been doing this for a long time, and so have others. And so have some of our pharma customers too. But it's very nice that it's called out, but not so explicit that it's almost impossible to finish. So I'm going to move to case studies now, everybody's favorite part, including mine. The first case that I'm going to talk about is a solute carrier transporter inhibition one. The client had already looked at the ABC, so we excluded PGP and BCRP. So all we were trying to do was look at the substrate and inhibition potential of this compound against this subset of solute carrier proteins. And this one's quite old, so the list is fairly contained. We were looking in stably transfected HEC293 cells, so this is a simple monolayer assay. And the vast majority of the study just chugged along and did its thing, and we got data. But the OATP1B1 and 1B3 inhibition part was actually not that straightforward and was one of those cases where you've got to go back and scratch your head and think a little bit before you can draw some conclusions. So this is what the majority of the data looked like. We didn't see any renal transporter inhibition. So the graph at the top is just uptake versus concentration. And as the no solvent control here, and the 0 micromolar drug, they're about 250 picomole per mg. As we increase the concentration of drug, not a whole lot happens. One could say at the end, perhaps there's a little tail, but it doesn't really matter because the concentration range that we were looking at based on dose projections was only up to 30 micromolar. Not a lot happens, very interesting. Positive control data are beautiful. No inhibitor has great uptake. Inhibitor has no uptake, so everything's clean for all these 5 transporters. Everyone's happy, we chug along, this is a quick study. In the case of the hepatic transporters, 1B1 and 1B3, we did see some inhibition. People are less happy, but we understand. But what we see is as we increase the concentration, we start losing uptake, which makes complete sense if we're looking at inhibition. The challenge here comes later. Because if you look at the data at first pass, it's beautiful. So we've got these lovely IC50s. One is 11.2 micromolar, one is 6.2 micromolar. It looks fairly normal. The challenge becomes the next step. So in this case, the client asked us to go forward a little bit and look at some just static modeling. So we went forward and did some calculations. And here's the calculations that we typically do for transporters. They're all lined out in the guidances. They're pretty, that the variables themselves look complicated, but the basic gist is fairly simple. If the concentrate, for the ones we're going to look at are here, the FDA 2020 and the ICH 2022. For OATP 1B1, these are basically the same equations. They just look different. So the only real difference is that here you add the 1 and here you don't. But the concentration, the maximum concentration, the inhibitor of the liver of this inhibitor at the liver inlet over the IC50 or the Ki is greater than 0.1, then we would want to move forward with a little bit more work, whether that's some more dynamic modeling or complex modeling, or whether that's some clinical work. The intent is that if you get to 0.1, if you get above 0.1, you need to do some further work. Back when we did this, it was if it was greater than or equal to 1.1, adding in that one factor. So there's a bit of a challenge in this, and as we're reviewing the data, we find it. We were doing some of the prelim work for cytotox instability and nonspecific binding. And we incubated this compound for 30 minutes in the presence and absence of control cells. And what we saw was that in the no cells, we get about 75%-ish back, which is workable. In the control cells, we lose a ton. We actually get up to 44% adsorption. So we lose quite a lot of it just to control cells. We've got a highly non-specifically bound compound. This is sticky. So when we go ahead and look at the calculations using the nominal concentrations, we have a little bit of a complicating factor in here. So OATP1B3 is pretty clean. You know what you're going to do. You go, okay, well, the IC50 was 6.2. At the time, the cutoff was 1.17, which is greater than 1.1. So we have inhibition potential and we need to keep going. But the 1B3 is a little bit less cut out, and then it's 1.09. So what are we going to do with that? We're going to say, well, technically, it doesn't, but my goodness, it's very close. In this case, it's not too terrible because you can do 1B1 and 1B3 in the clinic fairly easily together. But, you know, it might not have been 1B1 and 1B3. However, if you take into account that around half of it's non-specifically bound, theoretically, those IC50 values could actually be half the calculated values if we're using a conservative approach. If we do the NSB correction, then we end up with the R value here is 1.1899, which is clearly greater than 1.1. So at this point, we would recommend that for further investigation too. So it really did make a difference to this in terms of clarity of understanding where you need to go next with your drug. Case 2 is a little bit different. This one's a little different because we were looking at a PGP transporter situation in CACO2 cells. And as I said a while back, we performed this one in a transwell format, which is pretty interesting. So a transwell format, for those of you that don't think about this every day, is in my head, this is like a little beaker with sort of a little membraney cup inside. And you grow the cells and they're happy in a monolayer on the bottom of the little cup, which is an insert. And what you do is you put drug on various sides, either in this big side or this smaller side, and you see if it crosses the membrane in the presence and absence of inhibitor for your transporter. If you look, this is important. The volume of this part right here, the big beaker, is 980 microliters. The volume inside the insert is only 200 microliters. So there's a big difference in surface area to volume ratio as you think about this particular assay format. So the donor side is where we're putting the drug. We don't do unidirectional, we do bidirectional because it really helps you be more confident in the data. And so it could be either side as the donor versus the receiver. But if we are looking at B to A, because of the way the cells are expressed, if it's going from B, the basal layer, to the apical, that is active transport. And if we're going A to B, that's passive permeability. And so we measure both so that we know what to do with the data, right? But that does dictate how we run the assay and what we have to do with the data at the end in order to get conclusions. So I'm going to talk about some math a little bit, because this one, it becomes relevant to go back to first principles. So if you're in the industry and you do this a lot, you're familiar with Fick's first law, hopefully, which basically says that the flux is equal to the permeability coefficient correction times the maximal intestinal concentration. When we actually take this a little bit further, and when we do our normal transwell assay data processing, we use some of these relationships to get an apparent permeability, a PAP. And if you look at the equations and you come back, you're basically going the volume in the receiver divided by the membrane surface area, that's important, and the donor concentration at time 0, multiplied by the change in receiver concentration over time gives you the P-app. Because the units for these things are the same, the concentration at time 0 and the change in concentration above, what happens when you're done is you cancel everything else out and you get per seconds. So these two in the circle cancel each other out in terms of units. You can change that if you don't have concentrations and look at it from a mass perspective, and it's the same thing. The ratio is going to cancel out the units, so you can use it just the same as you could use a concentration. And this is very useful on occasion. So in this equation below, we're doing the same thing, but we're saying what is the mass of material versus what's the concentration in each place. Becomes relevant later. Transwell assays really suffer from non-specific binding problems. If you have something that's non-specific binding, you're going to see it pretty markedly in a transwell assay because there's different surface area to volume ratio on the A and B sides. We talked about that a little bit. And if you're trying to do substrate work, it's really difficult because you have to be able to measure the compound to low levels to be able to make sense of it. So non-specific binding can be a real problem. So when we looked at the data here, we realized that the recovery in the absence of cells, so there's no biological material here, it's just the transwell insert. At the lower concentrations, 0.5 and 2.5, we only get 41.8 or 54.3 recovery. So that means that we lost up to 58% through just adsorption to the apparatus at the low concentrations, which is obviously a lot. If we look at the recovery in the apparatus plus the control cells and the presence of the control cells, it actually shows you another problem. So now our recovery in the control cells is very low in the A to B direction, all the way down to 41%. This can make conclusions pretty hard to draw because you don't actually know necessarily what your concentration was. So now all the further calculations that rely on that are slightly unreliable. So we've got high non-specific binding and there's a marked contribution from the cells. If we look at the PGP substrate data for this KCO2 assay, the way that we look at it is to calculate P-ups and efflux ratio. So the efflux ratios are basically telling us how much is moving across the membrane over time. I said we do bidirectional, so we have it in both directions, which means we can calculate our efflux ratio. So it's basically what we're doing is putting the B to A P-up over the A to B P-up and getting a ratio to tell us what is happening more, passive permeability versus active transport. And at the low concentration, which is 0.3, we see that in the absence of inhibitor, it's 1.99 efflux ratio. In the presence of inhibitor, we've got 1.52. The 2017 guidance, which is the one we were following at this time, highlighted that a drug is an in vitro PGP substrate if it has a net flux ratio or efflux ratio of greater than or equal to 2 in cells that express PGP, as is 1.99. So it's there, but it's right on the borderline. And if flux is inhibited by at least one known inhibitor at an appropriate concentration, as is inhibited, but it's a little bit. This is tough to know. And the way forward would depend on a few things. People's appetite for risk and ultimately whatever the patient is whatever the disease state of interest is, but it is really truly inconclusive. It's not an easy one that you can just go, okay, next step is this. So we recommended that we did a little bit more exploration and we went back to the math. There's a really nice paper, I put the reference in here, it's pretty old, it's 20 years old now, but it really lays out how all of this works. So if you are interested, go get it. It's a great read, it's not very long. But it has a suggestion of how to correct the data processing for this binding issue. Here's that mass equation for the mass balance that we talked about for apparent permeability. What you can do is correct it for the amount that got stuck in the cells. And this is the correction equation right here. And basically all you do is say what was the donor amount and how much is stuck in cells, which you can approximate. And when you do that, you get a slightly different PAP with a correction factor. And that is how we reprocessed all the data looking at it from this perspective. When we do that, we get much cleaner data and a much easier path forward. So we recalculated them, accounting for all that material that got stuck in our cell monolayer. And what we found when we did that was actually the efflux ratio is more like 1. So you really don't have a PGP substrate happening. This is pretty much passive permeability. The inhibitor makes no difference at all at the low concentration. The higher concentration, there is a little bit of a decrease and the efflux ratio is a little bit higher, but it's not close to 2. So the efflux ratio doesn't actually approximate 2 when you're looking and accounting for, when you're looking at it and accounting for the amount that got stuck in the cells. So this is pretty conclusive. Okay, we're done, move along. I'm going to move to case 3 now, which was it started out as just the sort of tier 1 Keiko2 permeability screen type thing. The others were closer to tier 2 and tier 3. This is a tier 1, so it was supposed to be quite easy. Supposed to be quick and easy. All we were trying to do is look at permeability in Keiko2. Just one compound concentration plus or minus Valspadar, the inhibitor. That was only a one time point. We did a quick short LCM-SMS method so that we could analyze for the drug itself. And we were just looking for normal endpoints, and we were incorporating high and low permeability controls. Because this was supposed to be a quick endeavor and fairly simple, the client provided minimal information. So we didn't have a structure going into this, which is a challenge for me. I always prefer to have information in case we need it, but it makes sense that that was their decision. So when we got the data, they were very, very odd. First of all, when we were looking at 10 micromolar compound, which is quite a lot of compound when we're looking at transporter work, we didn't have anything, any PAP at all, A to B or B to A, because there was nothing in the receiver samples. Okay, maybe it's not moving across the membrane. But the other thing that was really odd is that the recovery data are just ridiculous. It's quite bad, just A to B, 150 and 167. But when we look at the B to A recovery, it's nearly 400%. That doesn't make any sense. So I basically, we take an approach, if something looks really weird, we should probably go back and look at it. We're not going to just say, hey, client, here's your data, looks bad, even if it's an early stage sort of thing. So we did repeat it, but we got almost the exact same results again. Is it non-specific binding? Is that why nothing's in the receivers? No, because we've got really high recovery, so odds are low. So it so happens that today I'm moonlighting about transporters. I have done transporter work for a long time, and I talk about it and things, but my real background is as a mass spectroscopist and biotransformation scientist. And so sometimes, even today, I was in that group, I did MedID, and then I moved and became the leader of that, and then I moved into operations. So every now and again, even today, and it's been like 10 years, every now and again, the analytical team will see something weird and come and ask me questions and, say, what do you think? They're a great team. They're really high functioning. Their leader today, Seema, is fantastic. But sometimes you just want an extra brain. And one of the nice things about working in a CRO environment is you have that extra brain. There's lots of people who've been working in this before. Many of our people have really long tenure and a lot of experience. So they came to talk to me. And I said what I always say when something looks like this, you go for the easy. You sure you put it in there? And they said, well, we've run it twice. It's almost the same results. And I said, this is very strange. Can you go back and just do the assay again? And I asked someone to watch them. And they did. Nobody was very happy about it, but they did do it. And actually, they got exactly the same results again. And I said, okay, well, very clearly, it's not that we're doing something wrong. So let's look at it again. We've already pretty much ruled out non-specific binding. But at this point, I sort of said, well, I'm blind now because I don't have the structure. I can't tell you where to go next because I don't have the information. So we went back to the client, who of course is getting quite anxious at this point because we've done this assay 3 times, this easy permeability, and they still don't have any data. And we said, we'd love to help, but we need the structure. So they gave it to us. And when we got the structure, suddenly things started to make quite a lot of sense. And we proposed a hypothesis. If you look at this structure, which obviously I can't show you most of, there is an amide smack in the middle of this. So those can hydrolyze enzymatically. It's often slow, but not always, and non-enzymatically in KCO2 cells. Sorry, the non-enzymatic is the thing that's often slow with amide hydrolysis. Enzymatic can be all over the map. Either way, hydrolysis at this amide is very, very likely. If you did hydrolyze this amide, which is what I thought was happening, you would get 2 products. You would get this side, amide hydrolysis product A, with a nice free amine, which is every mass spectroscopist's happy dance moment. And you would get this carboxylic acid product B. The one with the amine makes you happy because you know you can see it with electrospray. It's going to ionize like crazy, most likely. So you can look at it by LCM-SMS. The other one is more of a hit and miss. Sometimes they ionize really well, especially as an adduct, but a lot of the time carboxylic acids are harder to find. But it doesn't matter because we can look for this particular reaction just by looking at one of the products. So we went off to explore this hypothesis. And the first thing we did was get a high-resolution product ion spectrum for the compound itself, because we know the structure now, so we can get some nice diagnostic fragment ions. And we were very pleased because all the fragments are on the side where we think we're going to be looking. And they all made sense. It was a very, very easy biotransformation assignment. Structure elucidation assignment, sorry. And we found that this identical spectrum was in the donor sample. So we're absolutely sure there is some of this in the donor samples. It doesn't make it to the receiver samples. The analysts had indeed put it in there. Then we looked for the product ion spectrum for product A. We did some targeted unknown analysis to see if we could find this product A. And we could. And we confirmed the hydrolysis product by mass accuracy and by fragmentation. So we were very confident that this is happening. Product A was also detected in the receiver samples. So at this point, we've confirmed that this hydrolysis product is happening, the hydrolysis reaction is happening, and we've confirmed that there is movement across the membrane. So we go back to the math. As we talked about quite a bit a minute ago, in normal transwell assay data processing, which is what we're doing transwell, you can use these concentrations to figure out your apparent permeability, but they cancel out units-wise. Basic dimensional analysis tells us actually that all units cancel except the centimeters per second, which we know. So what does that mean? How is that useful? Well, this is a relative assay, meaning that we correct something but we can correct an amount by the same amount somewhere else, by the amount of the same thing somewhere else. So the units of concentration actually don't matter at all as long as they're consistent. So technically, you can calculate a p-app without ever going back and calculating a concentration, using just an area ratio of analyte to internal standard of the hydrolysis product without any reference standard. You never have to do any true quantitation. We have a lot of relative assays in ADME, but for whatever reason, we typically go back and calibrate and do absolute quantitation. In this case, we can't because there is no standard reference standard of this material. Nobody's planned for this before. When we go back and correct the data, we see a very different story. So what we did was we made basically a metabolite ID style method, or the kind of method we would usually make in hepatocytes. So it's pretty long LC because that way you can separate out parents from metabolites with great confidence. We're using high resolution accurate mass spectrometry, give us just that little edge so we know exactly what we're looking at. And when we did this, we re-injected all the samples with this longer chromatographic method on this other instrument, and we monitored both the parent and hydrolysis product A. And so here are the data for both of them. Now that we've separated out hydrolysis product A very clearly from the parent, we see that the recovery is much more logical. It varies between around 76 to around 101. That's normal. That is not weird data. There's still no drug in the receiver compound, but we know why now, that in the receiver side, but we know why now because basically it all hydrolyzed before anything else happened. If we look at the amide hydrolysis product, though, this is where things get super interesting. The efflux ratio for the amide hydrolysis product is 2.4. And if you add the inhibitor, it drops all the way to 0.3. So what we were able to do was confirm the permeability results for the parent compound. We didn't see any permeability because basically there was none left, or hardly any. And we were able to evaluate the permeability characteristics of product A. So there is here some addition of function from active transport by PGP, but it's for the amide hydrolysis product A. So at that point, we're able to say we have active transport of the hydrolysis product. We know that. We also know that the parent compound has pretty intense hydrolytic instability. So that's a useful thing for the drug developer to know because they're going to need to consider that all the way through this program. And so our conclusion was that the compound in CACO2 rapidly hydrolyzed to product A. Product A required active transport for efflux across the CACO2 cells. And so we knew exactly what was happening in this allegedly simple assay that we'd now been working on for a few weeks. So with that, I'm going to talk about lessons learned and moving forward. The experimental factors that we talked about today are known to make a really big difference to interpreting data, particularly transported data as we looked at today. Practical factors of the assays are really important. You really have to consider them when you're troubleshooting. If the data look very normal, cool. But if they don't, you really have to go back to fundamentals and think about what you're actually doing. Even go to math if you need to, which we don't always think about. We're so used to just processing data. And the big lesson learned here, I think, is that cross-discipline knowledge and collaboration can really make a difference when you have a challenging compound. And so we always like to have a very collaborative approach with our clients. Let's talk about your compound. Let's talk about what you're worried about, what you've seen before. That helps us help the people that are trying to develop the drug. And then that does seem to be much more supported by the ICH-M12. There's a lot, it's a very collaboratively written, it's a very thoughtfully written guidance, and that's been really nice to see. So looking forward to a single regulatory guidance document to make all our lives easier is a wonderful thing that is happening very soon, and it should theoretically go live around the spring of 2024. And I'm looking forward to that. I do have to acknowledge a couple of people. Andrea used to be our drug transporter lead. She's still our subject matter expert, even though she does other things these days, but she taught me an awful lot about drug transporters. Rebecca and Lois were very instrumental in terms of study directing some of these studies, so it was actually them that did the work. And then Andy Rhodes and Chase McCoy were in the lab at the time. Andy was the lab analyst doing the assays, and Chase was the analytical chemist who came to me. And those people were very, very cooperative with me and didn't make a giant fuss when I made poor Andy, who's really good, go back and redo this assay, and had Chase watch her because he used to do transporter work. And then I do need to also acknowledge Melissa, because without her there wouldn't have been any slides in the first place. And I will move us on to Q&A. We have a poll question now, and it's about looking at the ICH-M12 guidance. Are you currently looking for that right now? And if not, when do you expect to adopt it?
So here's our final numbers.
Very interesting.
Looks like we've got some questions coming in, if you'd like me to go ahead and start with those.
Yeah, that'd be great. Thank you, Michael.
All right, so do you add FBS, BSA to your CACO2 assay?
So it's in, some of those things are in the buffer mix. We don't add additional. We typically don't try to adopt the protein sync approach. We have played with it a little bit, but it can really complicate things. So if we don't have to, we don't. Every now and again, we will walk that path, but it's more of a case by case, I would say. It's a good question.
Do you perform any pre-incubation before the main CACO2 assay?
Typically, we only do, well, so we do a lot of custom work, so it kind of depends. Typically, we wouldn't. Typically, we would make sure they're acclimated and then go forward. But it kind of depends what we're looking for in the KCO2 assay. So that's a little bit, you got to think about that one.
How do you deal with unstable compounds such as case 3 example and generating IC50 values for inhibition?
It's a great question. So it's actually, it's really difficult, is the truth. There are various ways you can adopt it. It kind of depends what test system you're in and how unstable we're talking. I mean, if it's like case 3 where basically there's none left, that is super hard. And so in our case, we probably would have had to go all the way back to principles and see if there was a way to stabilize it or try a different assay. I have worked on a pretty extensive project years ago where we tried, gosh, I don't even know how many stabilizers. There's a lot of options there. There's a lot of places to go, but it depends how much time and effort people are willing to put in it. Because for some people, just the fact that it's unstable like that means that they're going to stop with the compound. If it's the only compound they've got, they'll probably put more effort into it. But it's very hard. If you can just correct the data, then that's wonderful, but doesn't always work quite that simply, unfortunately.
Okay, I did my in vitro work a while ago to meet FDA guidance. Will I need to do it again for the ICH one?
It's a great question. So the answer is it depends. And what we typically say is have a look at your data, have a look at your package, see what you've got. There are some differences that could mean you may need to redo something. We actually have consultants that offer that as a service, and we also do some of it internally too, when people are asking for a specific study, for example. But it's about doing a gap analysis and seeing what the scientific justification is, and it really depends when you're planning to submit, because as of right now, the FDA guidance is in effect. So until the M12 becomes truly official, until it's truly final, then FDA is going to be fine anyway. If your planned submission is a little bit later, I would recommend doing some sort of gap analysis, whether you do it yourself or get somebody to do it, and just see if you're going to need to worry about it. But for right now, you can go ahead with what you have.
Okay. A lot of people test for nonspecific binding to only the apparatus. Why do you include the cells?
That's an interesting one. I've had this conversation before with a few people. So a lot of people do that, and I'm not saying it's wrong. In my head, the stickiest thing usually that you've got in your assay is the cells. So we had this long debate. I don't remember when. Well, it would have been 2017 when we kind of had to really face, okay, you are going to include this as baseline normal. So we all had this big conversation internally about, it's expensive, it's slower, it's going to be harder to compete. But I really felt like the science kind of demands you to. If you know if you know that you have a really sticky piece of your assay, it just makes sense to me to investigate that when you're investigating everything else. So that's where we landed.
Okay, it looks like that concludes all of the questions. So thank you for your presentation today, Joanna, and thank you to all of you for joining us.
Wonderful. Have a wonderful day.